Good morning. I am Dr. Ramchandran, Department of Microbiology, Dr. N.G.P. Arts and Science College, Coimbatore. Today we are going to discuss about pharmaceutical microbiology and the quality subject. The topic is who it is WHO Good Manufacturing Practices. It covers the Unit 1 in pharmaceutical microbiology and quality assurance. So in this video, we are going to see about WHO GMP. What is WHO GMP? Then principles of GMP, quality control, sanitation procedures in pharma industry, and sterilization procedures and their types is followed in the pharmaceutical industry. So, who GMP for pharmaceutical products? GMP is the part of quality assurance. A set of principles and procedures it included. Good manufacturing practices is that quality be tested into a batch of a product, but it must be built into each batch of a product during all stages of a manufacturing process. It is designed to minimize the risk involved in any pharmaceutical productions that cannot be eliminated through testing the final product. It means the product to be analyzed is properly tested and verify their quality once again. So there are some risk are involving in these manufacturing processes. That is the risk of contaminations. This contaminations is the major problem in the pharmaceutical manufacturing processes, and incorrect labels. So it leads a very major mistakes during the manufacturing processes. So the inappropriate label gives inappropriate details and wrong details about the products. So that's why the correct labels should be mentioned or pasted on the product. So insufficient or too much active ingredients. So it is also a risk in the pharmaceutical products manufacturing. So the good manufacturing practices is the set of regulations. It codes and guidelines for the manufacture of drug substances and drug products, medical devices, in vivo and in vitro diagnostic products and foods. So what is the importance of a GMP is the poor quality medicines may contain toxic substances that have been intentionally added. Medicines that contains little or none of the claimed ingredient will not have the intended therapeutic effect. So the interrelationship is existing between the quality assurance, good manufacturing practices and quality control. So here we are going to see about the interrelationship existing between these terms. The quality assurance, it is the sum total of the organized arrangements with the objective of ensuring that products will be of the quality required for their intended use. The GMP is the products are consistently manufactured to a quality appropriate to their intended use. The quality control is a part of GMP concerned with sampling, specification and testing, documentations and release procedures that are ensured necessary and relevant tests are performed. The product is released to only after asserting its quality. So there are 10 principles are followed during the GMP. What are they? So the first one is design and construct the facilities and equipments properly. So it includes the building management systems. So the manufacturing facility should possess all the uh, required facilities. It fulfill the guidelines of GMP. Then they follow written procedures and instructions. It is called SOP, Standard Operating Procedures. The documentations work. What all the analysis has been done during the manufacturing process, it has to be documented properly. Then validate work. The process and analysis are performed in validated equipment only, and the procedures have been validated properly. Okay. Then monitor facilities and equipments. So the facilities has to be monitored periodically or passively. So with a proper validated equipment or calibrated equipments. So if it is a facility is a sterile one, you have to monitor the environment in a proper procedures to follow. Okay. So the monitoring procedures is different in a different facilities. For a sterile facilities, we have to follow environmental monitoring. What are the raw materials monitoring for their microbial load and the quality of air to be checked periodically. So write the step-by-step operating procedures and work on instructions. The seventh one is design, development, and a demonstrate of job competence. Then the protect against the contaminations. So contamination is the major problem in the sterile facility. So in any way, we have to check it out the contaminations and we ban the contaminations route. Okay, in this way, we can ensure the standard of the product and control components and product related processes. The final one is contact plan and periodic audits. Then this slide, who good manufacturing practices for sterile pharmaceutical products. They are general considerations, quality control, sanitations, manufacture of sterile preparations, sterilization. Sterilizations is included terminal sterilizations, aseptic processing and sterilizations by filtrations, isolator technology, blow or fill or field technology, personal monitoring, promises, equipments, and the final one is finishing of sterile products. First one is general considerations. So these are general considerations of sterile preparation should be carried out in clean areas, entry to which should be through airlocks for personal and or for equipment and materials. Then the clean areas should be maintained to an appropriate standard of cleanness and supplied with the air that has passed through filters of the required efficiency. The various operations of component preparations, product preparations, filling and sterilization should be carried out in separate areas within the clean areas. So these areas are classified into 4 grades. The manufacturing operations are divided here into 2 categories. The first one is those where the product is terminally sterilized, and second one is those which are conducted aseptically at some or all stages. So this is the clean room structures where we can find the HEPA filter at the ceiling, and the coating of these wall and floors are specifically and a special coating is maintained and followed. So the door is a pressured one. So this is the clean room pictures. The class 100 represents the sterile nature of the room. The class 100 means per square meter of the area should contain 100 particles. The ceiling, wall and other materials can be generate the particles that will be shut down in the floor or it can be deposited on the wall. So that dust can be facilitate the microorganisms lodgement. So avoiding this activity, we are wrap up the special coating in the wall and ceiling, and the air enter into these rooms has been filtered by a HEPA filter. Okay, so in this way we can maintain the class 100 sterile room. The second one is a quality control. The sterility test is appropriate for checking the purity of the product. The sterility test applied to the finished product should be recorded as the last in a series of control measures by which sterility is assured. The test should be validated for the products concerned. The samples taken for sterility testing should be representative of the whole of the batch, but should in particular include samples taken from parts of the batch considered to be most at risk of contaminations. The sterility of the finished product is assured by validations of the sterilization cycle in the case of terminally sterilized products and by media simulations or media fill runs for aseptically processed products. So the use of rapid microbiological methods to obtain earlier results and the microbiologically quality, for example, of water, environment and batches to be considered. This is a clean room for sterility analysis where the laminar airflow is containing the sterility equipments. This is the canisters where we perform the sterility test and the media is poured into this canister. This sterility test is containing in a closed manner. So this is a media pouring room at clean room. It is a class 100 room. Then sanitations is a clean area is particularly important. The sanitations. There are disinfectant and detergents should be monitored for microbial contaminations. So most of the sanitations procedures are followed with the help of a Dettol, Savlon, Bacillocide, Virocils, and 70% of disinfectants can be used for sanitation purpose. So the same type of disinfectant should not follow through the year or through the time. It should be modified with the time. The manufacture of sterile preparations is grade A, grade B, grade C, and D is followed. So this is the container used for packing finished products. So the recommended limits for microbial contaminations are given here. So grade A should contain less than 1 particles. The settle plates monitoration, the settle plate activity should contain in a clean area less than 1. The contact plates are less than 1. Then glove print less than 1 CFU per glove. The grade B contains less than 10 CFU per cubic meter. The settle plate contains 5, contact plates 5, and glove print 5. Then the C and D grade contains more number of CFU, but in the glove print there should not specifications given here. So this is the air sampler equipments. So this is the instruments. With the help of these instruments, we can observe the air in a closed area. So this instrument has a facility to accommodate a plate. It's a media plate. So the air sucked by these instruments filtered with this instrument, and the air particles are settled down on the plate. So this plate will be incubated and absorb the CFU. Then the sterilizations, it can be achieved by the use of moist or dry heat, by irradiations with the ionizing radiations, by clean oxide, or by filtrations with the subsequent aseptic filling of sterile fine containers. Each method has its advantages and disadvantages. The microbial contamination of the starting material should be minimal, and their bioburden should be monitored before sterilization. The biological indicator should be considered only as an additional method of monitoring the sterilization process. So this is the autoclave instrument installed in a pharmaceutical industry. The terminal sterilizations, this is the process where the sterilizations are performed at the last stage of the manufacturing facility. So this is the final stage of sterilization. So sterilization by gases and fumigants. Some of the situations we cannot follow the autoclaving. At that conditions we can follow the alternative method of sterilizations, such as we can sterilize a room with the help of gases and fumigants. So this is the fumigation process performing at the clean area with the help of a fogger. Then aseptic processing and sterilization by filtrations. So in order to maintain the sterility of the components and the product during aseptic processing, careful attention needs to be given the environment, personnel, critical surfaces, container, closer sterilizations and transfer procedures. The maximum holding period of the product before filling into the final container and the sterilization filter. So certain solutions and liquids that cannot be sterilized in the final container can be filtered through a sterile filter of a nominal pore size 0.22 micron or less. So such filters can remove bacteria and molds, but not all viruses or mycoplasmas. Consideration should be given to complementing the filtration process with some degree of a heat treatment. Filtration alone is not considered sufficient when sterilization in the final container is possible. So then the isolator technology. So the isolator technology is used to minimize the human interventions in the processing area. It results the significant decrease in the risk of microbial contaminations, because the human being is the highest barrier of highest carrier of the microorganisms. So that's why they are limiting the number of personnel in the manufacturing facility or sterile area. So the transfer devices may vary from single door to double door designs to fully sealed systems incorporating sterilization mechanisms. The transfer of materials into and out of the unit is one of the greatest potential source of contaminations. When blow or fill or seal technology, the seal units are purpose-built machines in which in one continuous operations containers are formed from a thermoplastic granulate filled and then sealed all by the one automatic machines. Below fill seal equipment used for aseptic production, which is fitted with an effective grade A air shower, may be installed in at least grade C environment, provided that the grade A or B clothing is usable. So because of this special technology, particular attention should be paid to at least the following: equipment design and qualification, validation and reproducibility of a cleaning in place and sterilization in place. We can say it is shortly CIP and SIP. The background clean room environment in which the equipment is located, operator training and clothing, and intervention in the critical zone of the equipments, and which aseptic assembly period to commencements of filling the manufactured product. In the personnel, only the minimum number of personnel required should be present in clean areas. This is particularly important during aseptic process. All personnel employed in such areas should receive initial and regular training in disciplines relevant to the correct manufacture of sterile products, including hygiene and the basic elements of microbiology. High standards of personal hygiene and cleanliness are essential, and personnel involved in the manufacture of sterile preparations should be instructed to report any conditions that may cause the shedding of abnormal numbers of types of contaminants. Periods of health checks is required to maintain the health of the process. The outdoor clothing should not be brought into changing rooms ceiling to grade B and C rooms. For every worker in a grade A or B area, clean, sterile, protective garments should be provided at each workstations. Gloves should be regularly disinfected during operations. Mask and gloves should be changed at least every working session. Operators working in a grade A and B area should wear sanitized goggles. Wristwatches, cosmetics, and other ornamental jewelry should not be allowed in these critical areas. So the clothing required for each grade is as follows. Grade D, the hair and where relevant beard and mustache should be covered. Protective clothing and appropriate shoes or overshoes should be worn. Grade C, the hair and where relevant beard and mustache should be covered. A one-piece jumpsuit gathered at the wrist and with a high neck and appropriate shoes or overshoes should be worn. The clothing should set virtually no fibers or particulate matter. So grade A and B, entry of a personnel into grade A area should be minimized. Headgear should totally enclose the hair and wear relevant beard and mustache. One-piece jumpsuit gathered at the wrist and with a high neck should be worn. The headgear should be tucked into the neck of the suit. The face mask should be worn to prevent the shedding of droplets. Sterilized, non-powered gloves of appropriate materials and sterilized or disinfectant footwear should be worn. The trouser bottom should be tucked inside the footwear and garment sleeves into gloves. The protective clothing should set virtually no fiber or no particles should be generated during the period. It is manufacturing time. So this picture represents the garments for a different grades area in the pharma industry. So if you see the grade C area down, you can wear a boil shoot. Then grade B area should wear the head gloves and head gown and this shoot. So this is the overall pictures about the grade A area and B. So the grade A area become grade B when the personnel enter into the grade A. So the premises, all premises should as far as possible be designed to avoid the unnecessary entry of supervisory or control personnel. Grade A and B areas should be designed so that all operations can be observed from outside. In clean areas, all exposed surfaces should be smooth, impervious, and unbroken to minimize the shedding or accumulation of particles or microorganisms and to permit the repeated applications of disinfectants where useful. To reduce the accumulations of dust and to facilitate cleaning, there should be no uncleanable recesses and a minimum projecting ledges, shelves, cupboards, and equipments. Pipes and ducts and other utilities should be installed so that they do not create recesses, unsealed openings and surfaces that are difficult to remove. Sanitary pipes and fittings should be used and threaded over the should be avoided. So the changing rooms should be designed as airlocks. They should be flushed effectively with the filtered air. The use of a separate changing rooms for entering and leaving clean areas is desirable. In general, hand washing facilities should be provided only in the first stage of changing rooms. The changing rooms should be equipped with mirrors so that personnel can confirm the correct fit of garments before leaving the changing rooms. Airlock doors should not be opened simultaneously and interlocking systems should be followed. So the warning systems should be operated to indicate failure in the air supply because air is a must in the personnel. So the air will be contained only the air, not the other just for dust or other particles. So it has to be maintained in sterile forms. It has to be filtered with a series of filter. There is a 0.5 micron, 0.4 microns and zero point 0.2 microns. A series of filters should be set in the air filtration facility. So the failure in the filter systems, air supply systems should be monitored properly. The indicators of pressure differentials should be fitted between areas where this difference is important, and the pressure differentials should be regularly recorded and failure alarmed. The consideration should be given to restricting unnecessary access to critical filling areas, that is, for example, grade A filling areas. And the equipments, whenever possible, equipments used for processing sterile products should be chosen so that it can be effectively sterilized by steam or dry heat or other methods. So finishing of sterile products. So the container should be closed by appropriately validated methods. The filled containers of parental product should be inspected individually for exogenous contaminations or other defects. Operators during these inspections should pass regular eyesight check using personal corrective lenses. Where other methods of inspection are used, the process should be validated and the performance of the equipment checked. Results should be recorded. So in a nutshell, we have learned here that what is G.M.P and the principles of G.M.P, clean room design and maintenance, types of sanitations and types of sterilization and its control. Thank you.